It's great to see a lot of people interested in network analysis. So as Steve said, I will start by reviewing hierarchical clustering, which we didn't develop. It's a standard method that was developed a long time ago. And then I'll tell you about dynamic branch cutting, which is the part that we did develop. So here is what I'm going to talk about, a brief outline of my talk. I'll tell you what is clustering actually. Most of you are probably familiar with clustering in one form or another. But just to start out slowly, I'll do that. I'll just briefly mention various clustering methods that are out there. I'll describe hierarchical clustering in a little more detail. And then go to the cluster identification in hierarchical clustering trees that are actually a product of hierarchical clustering. And there's two main ways that you can identify clusters in a hierarchical clustering tree. The first simple way is what is known as fixed height tree cut. It's a simple method that's well known to most people who work with clustering, but it has certain very important limitations, and that's why we developed the adaptive height tree cut. So instead of the fixed height, this is a more flexible method, and I think Bin Zhang was the originator of the idea, and he coined the name dynamic tree cut, or maybe Steve did. I'm not sure. So what is clustering? Clustering is a generic name for a lot of methods, but what they all have in common is that they start with a pairwise dissimilarity matrix between a set of objects. So here's a dissimilarity matrix between what I call gene 1 through gene 10, since our applications will be mostly in genomics, but it could be any other objects. It could be people, it could be, so whatever objects you want to analyze. And in the matrix, you see numbers, color-coded numbers. And so where the number is high, it means that two objects are very distant. And where the number is low, like on the diagonal, for example, the two objects are very close. So, of course, the object with itself has zero distance. That's sort of the definition of dissimilarity. But the objects close to the diagonal have sort of low dissimilarity, say 0.5 here or 0.4. So that's the input for the clustering method. And the aim is to find clusters or groups of objects that are closely related according to the given dissimilarity measure. So for example, if you look closely, you would say that probably this block here is sort of a cluster because these four objects, genes 5, 6, 7, 8, are closer related than the other genes are. And then maybe here is another cluster, genes 1, 2, 3, 4, because they are also less distant, more closely related than the other objects are. So, if you only have 10 genes and someone gives you a matrix like this, you look at it and you say, okay, there's two clusters. There's one cluster here, one cluster there. But if you have 20,000 genes, it's not so easy. So you need some kind of a automated method to identify the clusters. Now, the output of a clustering method is then a cluster label. The cluster label could be a numeric, for example, it could be number 1, 2, 3, and so on. We like to use colors in addition to numbers because colors aid in visualization. So there's this block here, we assign the color blue to it, so it's a cluster that we call the blue cluster or blue module. And this here, this block would be the turquoise cluster or object, cluster or module. Now if you have never heard of clustering, which I think most people did, but if you never did, here is a very simple and just sort of a hand-waving example. If you take the people living in the great state of California or in whatever state you're coming from, and you take as dissimilarity the geographical distance of their homes, which would you get as the resulting clusters? Well, you would get groups of people who live close to one another, which means you would probably get as cluster the cities, the towns, the neighborhoods, and so on. Now, if you think about this example, you may ask yourself or experts, what do you do? How do you assign people who live outside of towns in sort of unincorporated areas? So, people who are somewhere out there in a cottage or on a farm, they're not really close, they don't really have close neighbors. What do you do? Well, there are three possible answers. First, you can create a separate cluster for each outlying object. So each person who lives on their own, you say, okay, you're a separate cluster. This is good for some applications, but not for others. The second possibility is that you look at the map and you say, well, the nearest town to where you live is this one, and that's where I'm going to assign you. And if you were a post office manager or post office person who is in charge of zip codes, you would do just that. You would say, well, we want each person to be assigned to the nearest post office, but we're not going to put a post office for every person who lives on their own, right? We're going to put a post office in a town or in a city, and we're going to assign the far living people to their nearest post office so that we don't have to carry the mail too far. Now, the third possibility is that you leave them unassigned, in quotes, which means you don't assign them to any cluster, any town, anything like that. Now, most clustering methods produce what is known as partition. Partition means that every object is assigned to a cluster. It doesn't matter how far it is from a cluster, but each object has to belong to a cluster. Now, so sometimes this is desirable, but in biomedical applications, this is often a bad idea. And so we as in the people who created WGCNA, which is what you're here for, we don't like to do that. We like to leave the objects that are far from everything else unassigned, so they're kind of on their own. They're not part of any of the clusters that we work with. Okay, so what can you do with clustering in biomedical research? You can do mainly two separate approaches that you can take. Because in biomedical research, when you have high throughput data, you have some patients that you collect the data from, and then you have a lot of variables that you collect. For example, if it's a high throughput gene expression data, you may have 20,000 probes measured for each patient. And so you can try to cluster the patients, and people have done this, and it's been very useful for discovery of subtypes in heterogeneous diseases, in cancer, and neurological diseases, and so on. Or you can flip the problem on its side, and instead of clustering patients, you can cluster the high throughput molecular phenotypes that you measure. So it could be gene expression, it could be methylation, it could be proteomic. You could even try to cluster genotype, although that's usually it's not as useful. So in this way of doing clustering, the clustering of the high throughput molecular phenotypes is really the core step, as Steve mentioned already, is really the core step of network analysis techniques, and in particular of the weighted gene co-expression network analysis that you're going to hear about this week. And it helps discovering transcriptional or methylation or whatever other type of high throughput data you study, the organization of the organism, so transcriptional organization of the genome, protein complexes, and so on. Okay, so let me now switch and start talking more about the clustering itself. There are many clustering methods. There are entire books and monographs written on clustering, so obviously we're not going to not even try to cover everything here. So I'm just going to throw some names out so you can say that you've heard about these methods. The simplest clustering method is known as k-means. Then there is a variant of it for more general applications called partitioning around medoids. There are self-organizing maps, model-based clustering approaches, and as I said, there's lots and lots of other methods. Hierarchical clustering is one of these, so it doesn't really stand apart. It sort of swims with all these other methods in sort of the same pond. What does set it apart is that it works well with high-dimensional data and provides very useful, although not perfect, visualization. And that makes it a very good method for exploratory data analysis in biomedical sciences. And it does not require specifying the number of clusters beforehand, which some of these other methods do. So you can start with the data without trying to specify anything. You run a generic method, and it produces a sort of a visualization, which I'm going to show you in just a little bit. And then you can decide from that visualization what are you going to do next. Okay? So if you want to identify clusters or modules using hierarchical clustering, it's really a two-step process. In the first step, you construct what is known as the hierarchical clustering tree or dendrogram. And on the right here is an example of a hierarchical clustering tree, and it's actually an example derived from the dissimilarity matrix that I showed you before. And so this tree provides information on how objects are merged, and I'm going to go through that in a more detail in just a little bit. But in the second step, you basically have to identify branches that correspond to clusters in the tree. So this would be one branch, and this is another branch. And then, like I said before, you label the branches or the clusters by either numbers or colors or both. And in this visualization of the hierarchical clustering tree, we like to show the clusters below the actual tree. And when we do that, we mean that this blue color is assigned to this cluster. So all these four objects are in the blue cluster, and these four objects are in the turquoise cluster, and these two objects here, the gene 9 and gene 10, they're gray, and the gray color we reserve for the objects that are not assigned to any of the clusters. So they're sort of on their own. They're too far from anything. Okay, so next I'm going to show you a cartoon or a simple example of how the hierarchical clustering tree is built. So here's the dissimilarity matrix again. It's the same matrix that I showed you before, except I left out the numbers because you wouldn't be able to read them anyway. The matrix is too small. And on the diagonal, I just put gray colors. And the gray color means that these objects form clusters at this point, or branches at this point of the clustering. So what you start with is each object in its own separate cluster. And we start merging them now. So what do we do? So in step 1, you find the two closest objects according to the dissimilarity. So basically you find that the most red entry of this matrix, or the highest number in the matrix, and those would be these two blue numbers, these two blue objects, the genes, and you merge them. And the merging height here corresponds to the number that stood between the two. And you merge them, so now this gray area is a branch. Okay. Next step, you find the two nearest objects from among these 9. So there are 8 original objects and 1 composite object. And you're simply looking for the shortest distance between either two sort of regular objects or a regular object and a composite object. Okay, and it turns out that these two are now the closest ones, so you merge them. And again, the merging height here is the number that was between the two. Okay, so now we have two small branches and 6 leftover individual objects. In the next step, we again find the two closest objects, which is either the single objects or the composite objects. And the shortest distance now turns out to be between this gene here, gene 7, and this composite objects that has gene 5 and 6. Okay, so we merge them. So we add the gene 7 to gene 5 and gene 6. And the merging height is a little higher now because the distances sort of grow longer and longer as we go on. Next closest distance is between gene 3 and genes 1, 2. This blue part here. So we merge them. Okay? And notice that because we always find the closest distance and we merge the two objects, the next merge will be higher because it's a further distance. So we grow the tree from the bottom up this way. Okay, so we go on. The next closest distance is gene 8 with the composite objects of gene 5, 6, and 7. So we merge them. Next closest distance is gene 4 with this branch, gene 1, 2, and 3. So we merge them. Next one is gene 10 with genes 1, 2, 3, 4. So we merge them. And then the next one is actually these two composite objects are now the closest two objects. So we merge them. Okay, so we add this merge between them. And the last merge is then the gene 9. Okay, so here the clustering ends. We have a complete tree, complete in the sense that all objects are already on the tree. So starting from this similarity matrix, or distance matrix if you wish, you end up with a clustering tree that represents in some way the matrix. Okay, now at this point I want to just slow down a little bit, and maybe as you were following how I was following up with the merges, you may have asked yourself, well, so you know how to measure a distance between two single objects. That's the number that's between them in the matrix. But what do you do when you're supposed to measure a distance between, say, this object and this this single object and this composite object? And the way there are various ways of measuring the distance, and the various ways sort of give rise to different flavors of hierarchical clustering. We in our work like to use average linkage, which means that the dissimilarity between two objects, for example, this one and this composite object, is simply the average of all the dissimilarities between the individual single objects or singletons that make out the composite objects. There are other versions, for example, single linkage, where you take the minimum dissimilarity. There is what is known the complete linkage, where you take the maximum. So if you were to measure the dissimilarity between, say here, gene 8 and gene 5, 6, 7, the average linkage is you average these three numbers. The single linkage is you take the minimum dissimilarity, so you take the most red one, the closest one. Complete linkage is you take the maximum, so that would be this one. That's the lightest color, the furthest distance. And there are some other choices available that I'm not going to talk about. If you're really interested, there is a lot of literature. There's a lot of literature about which one to choose for which application, and of course, people have their own favorite choices. So, entire papers were written about the comparison between this and that, and how this is better, and then someone else says no, this is better. So, anyway, I don't want to go too much into that. Instead, let me tell you a little bit about how do you use hierarchical clustering in R. Hierarchical clustering is implemented in the function hclust, which is in the package stats, and it's a standard package, so you don't have to say library stats before you use hierarchical clustering. The function has two important arguments: the distance or dissimilarity structure, which is the matrix with the red and white colors, and the method, which tells the function which version of hierarchical clustering to use. And as I mentioned, we usually use average, but there are other choices available. And the result of the function is the hierarchical clustering tree. So the function gives you this output. Of course, it doesn't give you this output in a picture version. It gives you a structure that can be used can be displayed using the function plot, or you can use it as input to other functions that work with hierarchical clustering dendrograms. So that was the first step, how to build the dendrogram, the clustering tree. The second step is defining or identifying clusters from the tree. And this process is variously known as tree cutting or branch pruning. If you want to be more ecologically conscious, you can also just say cluster identification. I guess in modern times, tree cutting is not the most fashionable term to use. So anyway, if you look at these two pictures, like I said, this is a simple distance matrix of only 10 genes. The visual impression is that there are two clusters. One cluster is this rectangle here, or square. The other cluster may be this square here. Now on the tree, that sort of corresponds to two branches. Here is the branch of genes 1, 2, 3, 4, and 10 is sort of just barely part of the branch. And then this square here represents or corresponds to this branch 5, 6, 7, 8. And the gene 9 is sort of just above, just away from both of these branches. So what methods can you use to sort of identify these branches automatically? So for a simple tree like this, there is a simple solution, which is the fixed constant height tree cut. And it works like this. You pick a suitable constant height. So in this case, we pick a height 0.97, and if you want, you can draw a red line at that height, so you know what's going to where the line is. And then you take scissors, or whatever you want, and you cut branches at that height. So you should literally imagine that you take scissors and you cut along the line, and whatever falls down as an individual branch is a cluster. So here would be the clusters that you obtained. Gene 9 is a cluster on its own. Then you have the clusters of genes 5, 6, 7, 8. Then gene 10 would be a separate cluster. And genes 1, 2, 3, 4 would be a cluster together. Okay. Now, in genomic applications, we usually like to enforce a minimum cluster size. We don't like clusters that only have one gene in them. That sort of goes back to the idea that, you know, if you have a person living on their own somewhere far out of a city, you don't call that a city or a town. You know, they're just separate. They're away from cities. So similarly in genomics, we like clusters of genes or other variables that are at least 10 or 15 or 20 members strong. So we enforce a minimum cluster size. If we see a branch like this, for example, whose size is less than the minimum cluster size, we call it unassigned. Okay. Now, the problem with this method is that the cut height must be chosen very carefully. If you move this red line down, let's say to here, you would lose gene 4. If you move it up a little bit, sort of beyond this black line here, what happens is these two clusters merge, and suddenly you only have one cluster. You don't have two clusters. So setting the cut height properly, especially in moderately complicated data, like for example gene expression data, really requires an intelligent operator behind the keyboard. And in general, each application will require a different cut height. So if you have one data set, you get your modules and you cut it nicely and you get what you like, then you get a validation data set and you cluster it again, you will have to tune the cut height again very carefully to get something that may resemble the original discovery data set. So this is a major disadvantage of the constant height tree cut. Despite that, it's been successfully used in genomic applications and in WGCNA. So here is an example. The data that this dendrogram was created from are human brain expression data. They were analyzed this way by Mike Oldham and collaborators in 2006, but the data are from earlier works. And so you can, this is a dendrogram of about 3,000 or 4,000 genes, I think. I don't recall the number exactly. You can see very distinct branches. The branches are assigned different colors, so they form different modules. And when you analyze the data, you find that these modules group together genes that are expressed in specific brain regions. So each module has an interpretation of representing a particular brain region. I'm not going to go into more detail about the application. I just wanted to show you that even though the constant height cut is not really all that, or requires careful use, it can be used effectively and it can lead to interesting results. Now in R, if you want to use the static tree cut, you can use WGCNA functions, cut tree static or cut tree static color, that are in turn based on the function cut tree that's part of the standard stats package. The difference between cut tree static and cut tree static color is that cut tree static returns numeric labels that start from 1 and go 2, 3, and so on, and the unassigned label is 0. That's just a convention. The color version of the function returns color labels instead. So what you would get as number 1 from cut tree static becomes literally the label turquoise. Number 2 is the label blue, and so on. There are other colors. And instead of number 0, you get the color gray for the unassigned objects. Excuse me. Both functions take as their input the hierarchical clustering tree. You have to specify the cut height, so that's where you as an intelligent operator come in. And you have to specify the minimum cluster size. And if you want to know more, as always, use help for cut tree static. You will get a bit more description. So much excuse me, about the constant height tree cut. It's a simple solution and it works well in some applications, but it does not always work for complicated trees. And here is a fairly complicated gene clustering tree that comes from adipose data from mice. And if you think that this branch and this branch are fairly separate and should be called different modules, but then you also see a branch up here and up here, and you think that these two branches should also be identified, then I invite you to try, but you will find that there is not really any constant height that can separate, for example, well, let's say these two branches, and it can also keep this branch mostly intact. Okay, so in this sense, the constant height tree cut is too restrictive. It just doesn't allow you to identify the modules that you visually see in the clustering tree. So the solution is to make the height cut adaptive, and as I mentioned before, we call it the dynamic tree cut. Here's the reference for it, and I'll show you the reference one more time at the end. So how does it work? The branches are followed from bottom to the top, so it sort of follows the same process that I showed you when I showed you how the hierarchical clustering tree is built up. And when two branches merge, they are evaluated using various shape criteria. If the branches meet the criteria for calling them separate modules or clusters, they are called separate. Otherwise, they are merged. So let me show you how it works on the simple example. If you remember, genes 1 and 2 were the first ones that got merged together, so you start a new branch right there. Then we had genes 5 and 6, that's starting another branch. And then we were adding objects to each of the branches. So, you built up the branches. So far, so good. There's nothing really going on. We were just building two branches here. But then the two branches merge. Okay, and this is where sort of the gist of the method lies. Because the question now is, are these branches separate enough to keep them as separate clusters, or are they not really separate, are they not really all that separate and we should merge them? So what the function does, it looks at several statistics. For example, so here's a slightly more complicated example. So take these two branches and they're built up, and then here they merge, and the question is, are they separate enough? And so the function looks at statistics like what we call this gap, which is the distance between sort of the few lowest merges at the bottom of the branch and where the two branches merge. So that would be, if the gap is big enough, then that means the clusters are separate enough. It also looks at whether the core, this lowest merged group is low enough, whether the branch sort of hangs low enough from the top of the tree. It also looks at the number of the genes on each branch. If the branch is too small, it will not call it a separate module. And so there's a few criteria like this, and if each of the two branches meet the criteria, if they're big enough, if they're deep enough, if they look separate enough, then they're called separate branches. Otherwise, they are merged. Okay. Now, one thing that that's sort of a bit of a I wouldn't say a problem, but it's something that the dendrogram doesn't do very well for you, is assigning these objects here. So the two branches merged, and you would say that these objects now should belong to either one of the two branches or should be unassigned. Okay, and the dendrogram itself does not tell you which of these two branches these objects are closer to. So you have to go back to the dissimilarity matrix and you have to look at that. And so what we do here is, for each object, we basically assign it to whichever these two branches are closer. So what ends up happening then is that you get two nice solid blocks of colors, or two nice solid modules here, but then above the merge of, so here's the merge of the two branches, above the merge, which is this area, you get sort of a bit of a mess. And this is something that a lot of people who use dynamic tree cut struggle with, because each of these objects is assigned to the nearest of these two clusters. But here I'm showing you two color bands. One is the simulated one, so this is simulated data, and so I know which cluster each of these two objects was simulated to belong to. And this is what the dynamic hybrid tree cut does. And you can see that the simulated data also kind of look like a mess. And so it's not a problem of sort of the dynamic tree cut that the assignment is a bit, looks jumbled. It's actually that the dendrogram does not really tell you very well how these objects relate to these two branches. And so to do that, so again, to assign them, we just look for what is the closest cluster, and this is similar to what partitioning around medoids does, and that's why we call this step of assigning each object to its nearest cluster the partitioning around medoid step, or PAM-like step. Okay, and basically this picture here is just to convince you that the jumbling of the colors here is not an artifact. It's not a bug. It's actually a feature. It leads to something that makes sense. Yes.
Well, when you're building a dendrogram, can't you encode the direction of the node by node flipping?
You could. That's an excellent question. Sorry?
Please repeat.
Okay. So the question is, I'm going to paraphrase it. When you build the dendrogram from the bottom up, you basically, when you plot the dendrogram, not really when you build it, when you plot the dendrogram, you have a choice. At each merge, so each merge has two either objects or branches on the two arms of it, and you can flip them. And when you plot the dendrogram, the R function that does the plotting chooses the ordering so that the branches kind of look nice like this. But you could choose a different ordering, and in this case, you could choose an ordering where it would, I mean, the dendrogram would look kind of ugly when plotted, but it would make the colors go in single blocks. So that's an excellent point. Unfortunately, I mean, the plotting functions don't do it, so we could do it if we wanted to. The other point is that if you have a more complicated example, and there would be another branch here, so you would have 3 branches, then you could have possibly three different colors here. And so since for each merge you only have 2 options, but you may have to satisfy 3 requirements, you may not have enough. You may not be able to do it by simple flipping. So you would still get sort of jumbled colors at the bottom. Okay. So again, if you use dynamic tree cut, you may see jumbled colors like this where 2 branches have merged, but it's not a bug, it's a feature. Just remember that. Okay, in R, when you want to use dynamic tree cut, you have to load the package called dynamic tree cut. And the main function that does the tree cutting for you is called cut tree dynamic. And I encourage you, before you use it, to type help cut tree dynamic, because it has a lot of arguments. But the main input is the clustering tree, the dissimilarity matrix that was used to produce the tree. And then there are multiple options that fine-tune the cluster criteria and how the colors, how the branches are either merged or not merged, and how the individual objects are assigned to the nearest branch. But there are 2 most important options that modify the behavior of the function in the most dramatic way. The first one is deep split, and it's a numeric variable that can take values between 0 and 4, and basically controls how finally the clusters will be split. And the second important option is PAM stage, which is false or true, and it turns the assignment of clusters to its nearest object off or on. So let me show you a couple examples of how these options work. Here's a clustering tree of 3,600 genes from mouse liver data. It's not very important what the data come from, but it is a real data set, and it's a relatively complicated tree. And so if you set deep split to 0 and you don't use the PAM stage, you will get relatively large clusters. Most of the clusters will be relatively large, so there's not that much splitting. The cluster colors are very clean because there's no PAM stage to sort of jumble them up. But you will have relatively large blocks of gray color where there's unassigned genes, even though you may think that there are some branches here. So you know, you're missing a little bit if you set deep split to 0 and you don't use the PAM stage. Now if you increase deep split, you will get progressively more and more clusters, so more fine and fine splitting. So in general, more and more genes will be assigned to a cluster, but you're still missing blocks of, there are still blocks of gray color, which are these unassigned genes, which, you know, is sometimes not such a good idea. So what happens if when you do turn on the PAM stage, it's optional, it's by default enabled. So here's an example of deep split equal 2. That's the middle, sort of middle and default deep split. So you get, you know, a nice set of clean modules. But here there's a lot of genes that may belong to a module, but they're not assigned because there's 2 modules that merged before, and that's above a merge. And so if you enable deep split, then you get these sort of little areas of where the colors alternate really quickly, where these genes are assigned to its nearest cluster, which can be either this black or it could also be this green one here. There's some blue lines actually, I think, so they go to this cluster and so on. So the disadvantage of the PAM stage is, again, that the dendrogram is more difficult to interpret, but the advantage is that more genes are assigned to clusters. Okay, so I'm just going to summarize the highlights of this part. So hierarchical clustering is a useful method for finding groups of similar objects in high-dimensional data. It produces a clustering tree that's very suitable for visualization. The tree has branches, and at least the big branches, are the clusters that we're looking for. The cluster identification is also known as tree cutting or branch pruning. There's various names for it. The simple methods for cluster identification work well in simple situations, but they're not always suitable, especially in complicated clustering trees. And so that's why we developed the dynamic tree cut, because it's capable of identifying clusters in really complicated situations. Its most two important arguments that you should remember are the deep split and PAM stage. And the good thing about this method is that a single setting, which basically we use the default setting, works pretty well in a wide variety of situations, and it produces comparable results in many applications, which is good because that means that the method is suitable for automation. It basically doesn't require you to very carefully draw lines for a hierarchical clustering dendrogram and then decide which line gives you the modules that you like. It also has limitations. So starting with the clustering itself, it is a heuristic method. So it does not optimize a cost or penalty function, which is something that some people hold against hierarchical clustering. It is not a stable method in the sense that relatively small changes in the data can produce different trees, at least trees that look very different, and sort of give you somewhat different modules. Now, the visualization that the tree produces is not perfect, and this is true for all methods that try to visualize a high-dimensional data set in 1 or 2 dimensions. And users should not rely too much on the dendrogram. So I have shown you that, excuse me, that when you use the PAM stage, the colors under the dendrogram seem to be jumbled up, but actually they correspond fairly well to the modules that were simulated. So it's really a limitation of the hierarchical tree as a visualization method. And the dynamic tree cut, it works pretty well, but it also uses heuristic criteria, and the heuristic criteria are by no means unique. You could develop your own set of criteria and use them. So that's also sort of a limitation. It's you could have many different dynamic tree cuts, and they would give you somewhat different results. So you also have to be sort of careful and think about what you want to do and how you want to do it. The usage in R that you should remember is for the hierarchical clustering, you have the function hclust. For the constant height tree cut, the functions cutTreeStatic and cutTreeStaticColor. And for the dynamic tree cut, you use the function cutTreeDynamic. And please use the help command for all of these to learn more about the arguments, the output, and so on. And if you're interested in further reading, I'll point you to this article where the dynamic tree cut was described, and to the web page where you can see several examples and some more in-depth reading.
Thank you very much.